Hello, everyone. And for those of you new to MPN Voice webinars, I'm Nona Baker, co-chair of MPN Voice and an MPN patient. I'm delighted to know how many people have joined us tonight because we've got a very exciting webinar ahead of us with the updates and latest research from the ASH meeting. The ASH is the American Hematology Society. And it's wonderful that the professor is cross.
Hello, we need some people to mute if possible. We can hear a bit of background noise. Apologies. Yeah. Thank you.
Okay, sorry about that. And so we're going to have a really, really exciting webinar because as patients, we're always keen to know about the latest research. And we're incredibly lucky to have Professor Claire Harrison and Professor Adam Mead, who are going to give us the latest updates, which is a real treat for all of us patients. Thank you. Just a little housekeeping. I haven't got the slide. Okay. I'm sorry about this. There's a little technical problem. I need the slide a little bit bigger, Kanchan, please.
If we possibly go back to the previous view and just stick with that, I think that's probably better. There we go. Perfect.
Okay. The housekeeping slide, there's what we need now, Claire.
Got it?
Got it. Thank you. Just a few technical things in housekeeping. There is still a chance to submit your questions. We've got some really interesting questions that have already been sent in. Please use the Q&A box and when you submit your questions to the panel. And I just have to say that, you know, we're not going to be able to answer all of them, but we will do our best to answer as many as possible. But the one thing is the two professors can't answer individualized treatment plans. Those need to go back to your own consultants. The other thing that would be really useful for us is if you could fill out the feedback survey, which will be shared at the end of today's forum. So helpful for us because as we plan future webinars, we do want to meet the patient's needs. Okay. This is our agenda. We have the clinical updates from Professor Claire Harrison and then Professor Adam Mead. There will then be the panel Q&A. And then we will be closing at 5:45 this evening. I just want to talk to you a little bit about MPN Voice, because some of you who have logged into this webinar may not know too much about MPN Voice. MPN Voice serves the whole MPN community, patients and healthcare professionals alike. It started MPN Voice to provide accurate and up-to-date information, which is always medically verified. We support patients and their families with a raft of things. Up-to-date website. If any of you haven't seen our website, please log in. It's full of useful information. The forums, one like we're doing now online, and then our face-to-face forums that we are beginning to go around the UK as from May to invite patients to come and join local teams of doctors. That information will also be on the website. Online support, Health Unlocked, YouTube, Twitter, Facebook, Instagram, all with the latest news. Vlogcasts, those I do every month and have a conversation with somebody in the MPN community, which is then put up on YouTube. There's also a young person's blog. There are also CNS-led support groups. For those of you who live in America or Australia, those are nurse-led support groups within hospitals. We have buddies. Buddies are really important. If you're newly diagnosed, it's really quite often essential that you're able to talk to somebody who's been there, has been through what you're going through. And so anybody wanting a buddy, contact info@mpnvoice.org.uk. Advocacy, really, again, another really important and exciting thing, having a global patient voice in decision-making processes, including pharma and government, and also the process of drug approval and availability. We also have printed information, booklets and resources, and we fund research projects. This is just a snapshot. There is so much exciting things going on in MPN Voice. I do urge you to go to the website. Thank you. Now I'd like to introduce you to Professor Claire Harrison, who is going to do the first presentation on the latest research updates. Thank you, Claire.
Thank you very much, Nona. I was just concentrating so much on sharing my slides that I didn't update my microphone. So Adam and I are going to do a double presentation today about updates from the American Society of Hematology meeting and a few slightly broader comments as well. I'm going to focus mainly on clinical practice and Adam is going to focus on scientific updates, but there are some overlaps, of course, and where clinical work and scientific work comes together. And this is just really a sort of UK centric, but could be scaled elsewhere slide, actually, which was made by Beth Sala, who is a colleague of Adam's, who introduced one of the most exciting pieces of information that I'm sure you're going to be asking us questions about. So this sort of basically talks about MPNs, the schema of these diseases, ranging from ET and PV through to myelofibrosis and small percentage of patients who develop AML. We estimate that there are about 60,000 patients living in the UK with an MPN. And we think, just in summary on this slide, that we need new therapies that address, one, risk of transformation. Better ways of assessing these therapies are also required. So I'm going to talk about new impacts for old therapy for polycythemia vera, potential of molecular monitoring, and I see some of you already submitted questions on that, so we'll be partially answering that, but also coming back to that in our panel discussion, potential new treatments for ET, new options for upfront therapies for myelofibrosis, and new options that will come to the UK, but they will also come to other countries, and some of you will already have them in your country already. So one of the studies I cheekily want to talk to you about, first of all, is one actually that we've been running in the UK in collaboration with, across our whole network, funded to some extent by Blood Cancer UK, but also supported by MPN Voice. And you can see all of these different institutions, and indeed Novartis were involved in supporting us with this study and providing free ruxolitinib. And here we're looking at patients with PV, which this audience knows is caused by mutations in JAK2, characterized by clotting events. And our current treatment focus is on lowering risk of clotting with venesection, that's removing pints of blood, low dose aspirin, and in some selected higher risk patients, hydroxycarbamide or interferon.
Claire?
Sorry, Claire.
Yes.
Your slides aren't moving for some reason.
Oh, aren't they? Okay.
I don't know, sometimes you get this glitch with Zoom, but
okay.
I don't know if it's the
Bit
same
weird.
for everyone.
Shall I share the slides?
Yes, do you want to?
Yeah.
Yeah, let's do that. And I'll go on talking about PV. Thanks for giving me the heads up, Adam.
Yeah.
It would have been a bit sad if we'd got to like slide 30 and they weren't moving, wouldn't it? Yeah. Right, so Kanchan, let's keep, I can try to take control of them actually.
Yeah.
I don't think I can control them. I've tried. So can you move on and on and on again? And on again. Okay, so in our study, if we just stop here for now, we were looking at ruxolitinib, which is approved already, thank you, just stop there, for PV, but we can't currently prescribe it for our patients in the UK. So those of you who are joining us from other countries, you maybe can have ruxolitinib prescribed for you, but for PV patients we can't in the UK. So we started doing this study, and the idea was that our patients would be able to get access to the drug, but also we would be able to follow up patients for a long time, in fact for 5 years, without doing a crossover. And there was a crossover between the arms of the original trials with ruxolitinib Can you move to the next slide, please? On the next slide, you can see at the top is the study scheme where we compared ruxolitinib in patients who were not doing well with standard therapies to what we call best available therapy, which included hydroxycarbamide. And we followed these patients up without crossing over for 5 years. And we checked, first of all, could we control their blood count? Did they get blood clotting or bleeding events? Did they have transformation events? What happened to their quality of life? And we also checked what we called molecular response. And if you can see in the inserts here that ruxolitinib was superior to best available therapy in terms of controlling blood counts, but we also found, and this was a completely novel finding, that if we looked at patients who had clotting, bleeding and transformation, there were fewer of those events for ruxolitinib treated patients. And then when we looked separately at whether the patients had control of their blood count, regardless of what drug they were taking, we found that that was also important, and that controlling the blood count also was linked to less risk of bleeding, thrombosis, transformation, etc. So that it wasn't just the therapy, it was also that the therapy was working. So those things were important because they actually were not previously known, and we've always had arguments in the field about how important is it to control the white cell count and the platelet count as well as the red cell count. And then we went on and we had a big group doing that, supported by colleagues in Cambridge, by Adam, by colleagues at Guy's and St Thomas's. And we looked at patients whether they got a 50% reduction in the amount of JAK2 they had, which is called the JAK2 VAF, or variant allele frequency. So we said, if a patient had a 50% reduction in the amount of JAK2 they had, then they had a molecular response. And we found that at 1 year, those patients who had that 50% reduction, they also had a lower risk of bleeding, clotting and transformation. But that reduction was pretty much the same as the reduction we saw when they got good control of their blood counts, and also with ruxolitinib. If you look at the 3 graphs, they all look pretty similar. We then asked the question, if you look later on, so we were taking samples over the course of the 5 years, those patients who got a molecular response and they continued to have it, first of all, that was more likely with ruxolitinib. And secondly, and importantly, this went on and on correlating with reduced risk of thrombosis, hemorrhage, but also correlated with patients living longer. So these were important findings in terms of just basic principles of treatment, but also in terms of showing benefits for ruxolitinib for our patients. Next slide. This shows you a bit more information about molecular analysis. You can see on what's called a waterfall plot, which is the multicolored line plot on the top right, that the patients who went below the line had a reduction in their allele frequency, and the ruxolitinib treated patients are in blue. That we saw that for patients who had added mutations not in JAK2, that it was more difficult for them to enter a molecular response. And then Jennifer O'Sullivan, whose picture's on the bottom left, in Adam's lab using some amazing technology that he invented, correct, Adam? That the 3 patients who had a deep molecular response also had these changes in their stem cells, so suggesting that these were not just changes happening in the circulating blood, but also in the stem cells. Next slide. So that's a bit of science that I'm taking a bit from Adam. Next slide. So I'm working from an older deck here. This shows you about adverse events. So if you keep clicking, Kanchan, we'll click through a few builds here, but we saw us keep clicking. Slight increase in risk of infections with ruxolitinib, which we would expect, keep clicking. But we didn't see really differences in risk of myelofibrosis or acute leukemia, keep clicking. And then we asked a question about squamous cell cancers and cancers of the skin, which were much more common for ruxolitinib treated patients, which we know about as a side effect. But one more click, please. We saw that other cancers were not increased with ruxolitinib because we know this drug suppresses the immune system. Next slide, please. So in summary, this study is really important because it answers questions about standard practice. It starts to answer questions about whether molecular response is important. And we will be submitting this data, or we have submitted this data into the UK body which regulates whether drugs can be funded or not, which is called NICE. And there will be a hearing in May. Next slide. Whether this means that we should take ruxolitinib further forward, whether it means we should be doing molecular screening for everybody, is still a little bit up for discussion, because, for example, we don't know whether it was more important to have control of your blood count, even if you didn't have control of molecular response, or vice versa. And in the UK at the moment, and in France, for patients who have newly diagnosed PV, we are asking a similar question. So we're asking it earlier in the treatment, and that's a study called Mithridate, which Adam and I are both leading. Next slide. These are all the sites and all the investigators that were part of the MAGIC study. And if there are patients on this call, next slide please, who were involved with MAGIC, thank you so very much for taking part. If you scroll across these slides then please, Kanchan, there were several studies presented at the ASH meeting, next slide please, which also showed similar data, basically correlating reduction in variant allele frequency to better outcome for patient. This is one such study which was presented by an Italian group from Alessandro Vanucci and Paolo Guglielmelli, who basically linked reduction in a variant allele frequency with less risk of myelofibrosis. Asking the question, should we immediately change our practice on this slide? No, I don't think so. It needs careful thought and guidance. And this was done in a very carefully controlled way in one laboratory. So we would need to think about how we could coordinate that and really understand it. But those kind of questions are being answered currently. Next slide, please. And there were other data also showing similar information. Next slide. And next slide. So what about ET? So a couple of new options are coming for patients with ET and in clinical trials. And some of the data that was shown at ASH involved Imago, the Imago drug bomedemstat, or IMG 7289, which inhibits a molecule known as LSD1. And in the updated information from this study, we saw good control of the platelet count in 100% of patients treated for more than 24 weeks. And this occurred regardless of whether they had a JAK2 or a CALR mutation. Next slide. And we saw that the white cell count remained stable or normalized, and the hemoglobin was maintained, which is important because many of our treatments do cause anemia. For example, in the MAGIC ET study, ruxolitinib caused a lot of anemia. What I'm highlighting on the right was that fatigue was very much improved by this drug. And as we know that many of our patients are affected by fatigue, and so if it is true, and if it shows out in what we call a randomized study, which is what the phase 3 trial is, that fatigue is improved, then this will be important. A study with this drug in PV is going to open in the next few months in the UK. Next slide, please. Now, what about data with regard to myelofibrosis? That's coming next, but I just want to highlight that with this drug, in the MANIFEST study, the drug is called pelabresib. This drug is also being looked at in ET patients, and we're expecting to see some more information in the summer. But what I'm going to show you in the next few slides are some of the data with this drug in myelofibrosis. Next slide. And one of the questions we have currently in the field is whether we can do better than just using a JAK inhibitor alone. So at the ASH meeting, we saw data for ruxolitinib with PEG interferon, and from 2 studies with navitoclax and pelabresib. There is a third drug, parsaclisib, which was being studied in this setting, but those studies have just recently been stopped. One of the questions we have with these new combinations is how will we define enough benefit for patients? Will it be better spleen response? Is it better symptom responses? or is it that you get better molecular response or reduction in fibrosis? So these are all open questions. There's a question in the chat. The name of the drug was bomedemstat for ET. Next slide, please. So these are the candidates for things that we are looking at to try to understand if a combination is better than a monotherapy. Next slide. So I'm just going to quickly show you the data with ruxolitinib and pegylated interferon, and then the other two combinations. Next slide. This is the data that was presented from what's known as an academic study. The MAGIC-PV study is also an academic study. So here the trial is designed by clinicians or academics, and the endpoints are designed by them. Pharma sometimes involved because they will provide free drug, which was the case in MAGIC-PV, and also in this study where ruxolitinib with pegylated interferon alpha-2A was looked at in patients with MF. There were high rates of spleen reduction, which is shown in the what's called the waterfall plots, top left, that's the multicolored bars, and also allele burden reduction, especially for JAK2. Those patients who had additional mutations had less JAK2 molecular response. And interestingly, pegylated interferon is difficult for some patients to tolerate. Maybe patients on this call appreciate that. And what was described here was that quite high doses of interferon were able to be given and were better tolerated. They also looked at bone marrow picked images down the microscope and inflammatory cytokines, and we'll await some more data from this study. Next slide. Next, I'm going to show you the data for pelabresib, which is the BET inhibitor. Next slide, please. And here is the data for first line, so patients who haven't already had a drug. On the left, you can see that we saw reductions in JAK2. We also saw reductions in bone marrow fibrosis. And if you look on the right, there are some special stains. So the brown blobs, for want of a better way of explaining it here, are megakaryocytes or platelet producing cells. And these are usually closer together in patients with myelofibrosis. And what we found was that whilst the megakaryocytes moved further apart and became more normally distributed, it was more likely that patients had a spleen response. And that's what the fancy graphs on the left, which are called violin plots, are showing. So we did see some normalization of the bone marrow, which is also really important and interesting. Next slide. So that drug combination has been in a randomized study, which we're currently still conducting, and we've recruited all the patients to. The other drug combination which we need to watch out for this year is navitoclax, which inhibits the cell death or activates the cell death pathway in combination with ruxolitinib. Next slide. And what we saw in Europe and in the American Society meeting was some data for patients who haven't previously had a JAK inhibitor. We saw good reductions in spleen, and we also saw that about 50% of patients had a reduction in the amount of JAK2. And I already mentioned that those patients who had additional mutations sometimes didn't have reductions in their JAK2, but in this study with this combination, they saw that even if they had additional mutations, they did still get reductions in JAK2. So this study, this combination is also in an upfront study, which is fully recruited, and we're expecting results later this year. And there's also a study adding navitoclax to ruxolitinib for patients who have lost their response to ruxolitinib. That is still open. Next slide. All of these newer agents are all looking at what happens with bone marrow fibrosis. And there was one presentation at ASH where in a study comparing momelotinib, which is a different JAK inhibitor, to ruxolitinib, and looking at what happened to bone marrow fibrosis, they presented a lot of data which was quite controversial, showing that even if patients had a change in bone marrow fibrosis, it didn't correlate with spleen or symptoms or anemia benefit. Maybe this is just because it was too early, or maybe it's because of that it was a JAK inhibitor that was used to reduce the fibrosis grade. So I think the jury is still out there, but this is quite an important piece of data for us, and it's making us think about what we should be measuring and how. Next. So all of these different candidates for defining improved response are unvalidated, and at the moment we're still looking at spleen and symptoms, but we will be collecting all of this information going forward. Next slide. Adam will probably talk a bit more about this. So of all of these, the navitoclax, pelabresib, and will be available later this year in terms of results. And navitoclax is being tested in the second-line setting, and there are lots of other drugs, and there was lots of other data presented at ASH showing interesting preliminary results. Next slide. Some novel JAK inhibitors either are or will be available, just thinking about UK, and I'll touch on other countries later this year. Next slide. One is momelotinib. This looks like a bit like ruxolitinib. It inhibits JAK1 and JAK2, but if you look on the right-hand side, it also inhibits ACVR1 and also then reduces hepcidin and improves anemia in some patients. So this drug is available on what's called compassionate use basis, just becoming available at the present time. And we're expecting that it should be approved hopefully later this year in America and maybe also in Europe. Next slide. And there have been several phase 3 studies with this drug, most recently the Momentum study, which compared it to danazol, which is an anabolic steroid. And we know that it benefits spleen symptoms and anemia. And we also know it's safe to immediately transition between that drug and ruxolitinib. Next slide. And then also to touch on pacritinib, which is another JAK inhibitor, which mainly inhibits JAK2, not JAK1. But we also learned that it also inhibits the hepcidin pathway at the ASH meeting. That data was presented. Next slide. This drug is particularly focused on patients with platelets less than 50, and there is a study ongoing at the moment called the Pacifica study. Pacritinib is approved by the FDA, so if there are any listeners from America, and we know we have some registered, you may be able to already access this drug. We know that it can be given at a good intensity, so almost at full dose, even if patients' platelet counts are low. And compared to ruxolitinib, which is what's shown on the bottom right, we had better spleen and symptom responses. Next slide. And lastly, I think Adam's probably going to touch on this very specifically, but very excitingly, we did have a presentation talking about this antibody which recognises the mutant calreticulin, which is circulating and binds to the outside of stem cells and triggers platelet production by binding to the thrombopoietin receptor. So we can expect to see some studies with this kind of therapy. And in fact, we already have vaccination studies ongoing at the moment in the UK and America. Next slide. So these are some of our practical points which we shared with clinicians and would share with you today. That for PV patients, we learned that ruxolitinib has wider benefits. We had some tantalizing information about controlling the JAK2 VAF, but we also learned that really good control of the white count and platelet count and hematocrit is important. We've been encouraging our colleagues to think about this first-line study, Mithridate. So patients are eligible for that if they've got a diagnosis of high-risk PV and are within 15 years of diagnosis and 10 years or less of treatment. We touched on other trials that are currently available and also highlighted that momelotinib is available for compassionate use. So I'm going to hand straight over to you, Adam, at this point. I hope you have more luck sharing your screen than I did, and then we'll take some questions. And thanks, Adam, for joining straight after your clinic.
Yes.
In fact, I think you're still in your clinic, aren't you?
Kind of still in my clinic, yeah. Got all my paperwork to do, but there's no patients waiting.
There's always paperwork to do, isn't there?
Okay, let's see if I can get my my screen to share a step. Great. So you should
We can see your screen. I can't see it in slideshow view. And if people want to put questions, then they can put them in the chat. So now I can see your slides. I'll stop you if they don't progress, Adam.
Great, great. Okay, well, thanks very much. So thanks very much for the invitation to the MPN Voice team. And what I wanted to talk about is really following on from Claire's clinical talk, a deep dive into two particular studies that were presented at ASH that I thought were particularly exciting, and really linking in very nicely with what Claire was telling you about, just to really focus and think a little bit about what everyone is really excited about, I think, in the field at the moment, both scientifically and clinically, which is whether we can move our treatments towards something that we call disease modification in myeloproliferative neoplasms. So I want to talk a little bit about, in the next 15 minutes or so, about some exciting developments. Did the slide change?
Yes, it did.
Yes, it's moved.
Excellent. So I thought it's useful because there's a lot of talk about this that may get picked up in patient communities about disease modification and what does it actually mean? And, you know, to me, I've got quite a simple view on what disease modification means that I've tried to summarize in this little cartoon here. So if you think of blood cell production over on the left-hand side here, as in everyone, there's normal blood producing or hematopoietic blood producing stem cells that produce millions of blood cells every minute of every day throughout our whole life. And they're very clever cells that are very long-lived. And what happens in the development of myeloproliferative neoplasms, what we now know from some lovely work from the Sanger Institute and Jyoti Nangalia, that was presented at ASH a couple of years ago, is that following the acquisition of a mutation in JAK2 or cow reticulin for that matter, that occurs in a single stem cell that gradually, probably over many decades, evolves and expands to the development of an early myeloproliferative neoplasm. And in some patients, over the course of years, additional mutations are acquired that causes the disease to progress. So this is a very long-lasting process, all starting off from a single cell. And that is why sometimes you might hear this term, which is called clonal disease or clonal disorder. So myeloproliferative neoplasms are clonal diseases, and that means because essentially all of the myeloproliferative neoplasm cells are a clone, all originating from one cell. So back to the point about disease modification, what do we mean by that? Well, what I mean by that is, can we reverse this situation where you've got a mix of different mutations mixed in with a few normal blood stem cells? Can you use treatment to actually reverse the situation and get back to normal, healthy blood cell production? So eradicate the mutations. So a durable clonal response means getting rid of the abnormal clone and restoring normal blood cell production. And if you can do that, it seems obvious to me that this will correlate with all the other important clinical endpoints that are important for patients, you know, symptoms and burden of the disease and the risk of the disease progressing. And all the studies Claire was talking about this term variant allele frequency and molecular response, you'll see all these different terms. That is essentially measuring the number of cells in your blood cells that carry a particular mutation. So the molecular response is essentially a surrogate marker that we can use in the laboratory to measure this process. Are we reversing this situation and getting the blood cell production back to this situation? So that, in a nutshell, I think is what we're ultimately trying to achieve with disease modification. So, you know, that's what we're ultimately trying to achieve, but is this goal achievable? Well, Claire's already told you, we're really beginning to make really significant progress. And I wanted to talk about two studies that were presented at ASH in a bit of detail, just to tell you about some exciting developments that haven't quite made it to the clinic yet. So here's the first one. This was presented at the plenary session, ASH. This is the one that Claire just mentioned. So the plenary session basically means out of 5,000 pieces of scientific work submitted to the meeting as an abstract, it's selected out of the top 6. So really, really a lot of excitement because they're scored very highly and presented at this prominent plenary session. And so this is, I'm afraid there isn't a nice name for this particular treatment yet. So it's got this code name here that I won't read out, but it's an antibody treatment that is selectively targeted against mutant calreticulin in myeloproliferative neoplasms. And it's developed by a drug company in collaboration with some academic researchers, including some from the UK, for example, Ian Hitchcock, who is based in York and is a terrific scientist. So how does this work? So calreticulin mutations, many of you will have heard of. So they occur in about 30% of persons with essential thrombocythemia or myelofibrosis. And calreticulin is a protein, so the mutation in DNA leads to the production of a mutant or damaged form of the calreticulin protein. And then the important thing for you to realize that you may not know is that mutant calreticulin finds its way to the surface of cells. So it is expressed on the outside of the blood cells that are carrying the calreticulin mutation. And there are two particular recurrent types of calreticulin mutation that you may have heard of, one called a type 1 and one called a type 2 mutation. And what the folks at Insight wondered about is whether the presence of this mutant calreticulin on the cell surface might actually open up therapeutic opportunities, because this is a rare example of a disease-specific protein. So it's only expressed on the surface of the MPN clone of cells, not on the normal blood cells. So that might be a unique opportunity to actually target this using a therapeutic immunotherapy. So using the immune system to target the cells, in this case, using therapeutic antibodies against the mutant calreticulin. So that's what the folks at Insight developed with this catchy code name here. This is the classic shape of an antibody. So this indicates the antibody that is selectively binding this specific mutation in calreticulin. And they did a whole series of experiments that, there's a lot of jargon on these slides. Ignore all the details. I will explain what it means in as simple terms as I can. Here is a control antibody, so a non-specific antibody. This is the calreticulin antibody, and this is an in vitro, in laboratory test tube assay, showing how well the mutant calreticulin antibody controls the cell division and proliferation. It is very specific to mutant calreticulin, so it doesn't work for JAK2 mutations. It only works for the mutant calreticulin. They also showed the same thing in blood samples taken from patients, and that's shown in these slides. They even showed that it works against the stem cells taken from patients. Those are the disease propagating cells that underlie the disease development, and very, very substantially reduce the number of disease-associated megakaryocytes. I've got a picture of that in a minute. They also showed that this worked in an in vivo, so in an animal model, in a mouse model system. They showed that this antibody was highly effective at controlling platelet counts, normalizing the platelet counts compared to a control antibody. Very impressive. And here's what the bone marrow looks like in this mouse model. Lots of abnormal megakaryocytes. Those are the cells shown in brown. Those are the cells that produce the excess numbers of platelets in essential thrombocythemia and myelofibrosis. And this is the dramatic reversion of that appearance with the antibody treatment. Impressively, and relating to the disease modification I opened up with, it also has a very strong effect on suppressing the total number of mutant calreticulin stem cells. So it's inducing these clonal remissions that we're really trying to achieve in terms of disease modification. So incredibly exciting work, I think. This is moving to phase 1 clinical trials this year, specific for calreticulin mutation patients. And it's definitely one of the most exciting developments in the field for many years, in my view. Okay, then I want to present some work that actually was led by my colleague in Oxford, Beth Seiler. And in this work, which is summarized on this slide, what we tried to do is create what's called a cellular atlas of myelofibrosis. And to do that, we used a mouse model system of myelofibrosis, where the mice develop large spleens and they develop myelofibrosis. This is the similar model to what was used in the previous presentation. And what we set out to do here is really work out all the different types of cells that are disrupted in the bone marrow during the development of myelofibrosis. Surprisingly, that is not something that is known at this stage. So the exact cell types that cause myelofibrosis are really not so well characterized. There are some cells that have been implicated, for example, the megakaryocytes that I've mentioned before, but other cell types are much less clear. So here, importantly, we set out to characterize not only the blood cells, but also the support cells within the bone marrow. And I'll talk through this pretty quickly. What it uses to create this atlas is a single cell technology. So you take out millions of bone marrow and blood cells, and then you can analyze each cell individually for all of the genes. So it's a very kind of cutting edge, exciting technology. And then you can classify all the different cells. So each dot in this plot here is an individual cell that we've then mapped onto this plot here. And you can see all the different cell types in blood cell production. So red blood cells, white blood cells, stem cells, megakaryocytes, lymphocytes, so the immune cells, and these interesting population that I'll come back to, EBM cells. Those are eosinophils, basophils, and mast cells. They're normal blood cells, and rather unexpectedly, we found they were really strongly disrupted during the development of myelofibrosis. And we also characterize these disease-associated stromal cells, as they're called. So these are not blood cells, they're support cells in the bone marrow. And the crucial thing that we found was that there was this expansion of the eosinophil, basophil, mast cell populations associated with myelofibrosis, not something that was recognized before. And there's a strong expansion of these non-blood cells called mesenchymal stromal cells, again, not recognized before. So we're learning new things about the crucial cell types that might underlie the development of a severe blood condition like myelofibrosis. The crucial cell type for laying down fibrosis and scar tissue are these cells that are called fibroblasts, or FIB for short. And surprisingly, we found that they were not strongly altered in number in myelofibrosis, but what they did show in myelofibrosis was very strong inflammation-associated signatures. So the fibroblasts are not increased in number, but they're changed in their qualities that create this very inflammatory microenvironment. And the same is also true for these for the mast cells. So why is that important? Well, we can now interrogate these key cell populations, the fibroblasts, the mesenchymal stromal cells, the megakaryocytes, the mast cells, and say, is there anything that's common between all those different cell types that might be a target for therapy. And if you do that, you find just very few genes, actually, 20,000 genes in the genome, and we found just 2 that were strongly dysregulated. One of them, this very interesting molecule called galectin, which is a pro-inflammatory mediator. So it causes inflammation and it causes fibrosis. So we thought that was pretty interesting. We studied some patient samples and showed that in myelofibrosis, there is a huge increase in the amount of this molecule galectin compared to in healthy disease or in essential thrombocythemia. And then we could go back to the mouse model. And again, we developed a different antibody treatment and showed that this could very strongly reverse the size of the spleen, improve the blood counts, hematocrit and platelet count, and reverse the fibrosis. So we think that this looks a very promising therapeutic target in myeloproliferative neoplasms. And there are some therapeutic agents already moving towards clinical trials. Okay, I think Claire's already talked about the MAGIC study, so we've got plenty of time for questions. I'm going to skip over that. And the only thing I wanted to mention right at the end, to loop back to the disease modification point I opened up with, was this is the combination of treatments that Claire mentioned, ruxolitinib combined with navitoclax in patients with myelofibrosis. So the clinical trials in the second-line setting, and really exciting, in the first-line setting, there are patients showing these molecular responses really quickly after the initiation of the treatment, even within 12 weeks, patients showing a strong clonal reduction and molecular response. So this is really exciting. It's really not been seen before in myelofibrosis. And just to give you a flavor of why this might be so exciting, this is a preliminary analysis from one of these studies showing that in patients that did achieve this kind of molecular response, their outcomes were much better than patients who didn't manage to achieve this molecular response. So I think what we're going to see over the coming years, it's not kind of ready for prime time in terms of everyone having molecular monitoring, but I think it is coming that for many patients over the coming years, we'll be thinking about, can we use a treatment to achieve a molecular response and really suppress that abnormal clone of myeloproliferative blood cells. So thank you very much for listening. It's very exciting times in the field, and I hope some of that came across.
Very good, Adam. Thank you so much. Brilliant. I'm glad you had no problems moving your slides. They're very complicated. But as always, you put across really very complicated science so eloquently. Thank you so much. And thank you so much for everything that the teams in Oxford do scientifically. So we're going to do a Q&A now. Look, we've got lots of clapping hands going up on the screen there.
Claire, can I just say a huge thank you to both of you on behalf of all the MPN patients around. This has been so exciting and so enlightening and gives us all great hope for the future. Thank you both.
No problem. So I don't know, Adam, whether you can see the questions or whether I just get the opportunity to ask you all of them.
Yeah, you find the difficult ones at me.
Oh, so one of them, of course, we're really excited by this mutant calreticulin antibody, and we're trying very, very hard to be allowed to use that in the UK.
Yeah.
But there's a very nice question that's appeared in the chat from Paula Darwell-Stone. Is there an INCA 033989 equivalent for development for JAK2?
Yeah, and that is, that would be really lovely. But it's more difficult with JAK2. And the unique thing about the calreticulin is it's a protein that makes its way to the cell surface. That's what makes it very amenable to this type of approach. Unfortunately, with JAK2, JAK2 is a protein and mutated in a similar way, but it's held within the cell. It doesn't make it to the cell surface. But I don't think that means the situation is without hope. I think there are possible approaches to target cells specifically using these immune therapy types of approaches that have the JAK2 mutation. There's some very exciting research being carried out at Guy's Hospital with that Claire's involved with, and I've forgotten the name of the Italian chap that's leading that. But
you mean Michele,
yes,
Miele, and Sharam Kodashti, yes.
Yeah, and so that's very, very exciting, and it is possible that the JAK2 mutation also, Ian Hitchcock, who's a real expert in this, is also very interested in studying how JAK2 inside the cell might still make changes on the outside of the cell that could be targetable. So I think, that's further behind, but are people actively exploring this type of approach for JAK2? Definitely.
Yes, I would say that's definitely true. And in fact, I'm going straight from this meeting to a meeting about vaccination and looking at progress with the vaccination trial and others. But I also think one thing that's really interesting to point out is we discuss a lot interferon, and now we know about ruxolitinib reducing JAK2 allele burden, Adam. But it seems to mainly do that for JAK2, doesn't it?
Yeah.
Certainly. So, maybe we can target calreticulin with an antibody, and maybe JAK2 we can target with some of these other drugs and reduce the amount of variant allele. And I'm not quite sure why JAK2 seems to be a little bit more sensitive to interferon and to ruxolitinib. But any thoughts from you?
I'm not sure. I do know the answer to that. I'm not sure that really any studies have shed particular light on that. I still think it's possible that there is an immune element to the responses seen with interferon, although the current working hypothesis is that it's a little bit complicated to explain. But what we think might be happening is that in those really important blood-producing stem cells, when they've got a JAK2 mutation, they turn over a little bit faster than they normally would. And what interferon does is, it might sound surprising, but it makes them turn over even faster. And you might think that's not absolutely the opposite of what we want to achieve, but actually in stem cells, that can be very useful, because as stem cells turn over, they become depleted. And that's probably how the interferon is working with JAK2. And calreticulin is just a little bit less strong at causing that mutant stem cell turnover. So that might be why there's that differential effect, but that's very speculative.
Okay. Adam, there's a question in the chat about, I'm going to ask you this question, let you think about it, and then I'm going to answer another question while you're thinking about it. So this question comes from Vicky Ingram, and it's about, what would we need to do more individual molecular monitoring? Is it lab capacity, patient comfort, or is it budget? And then there's also a question about, why don't we tell patients their allele burden? So I just wonder if you could think about those, and I will answer in like one sentence a question that's come from Anne Jessamine about the MPL mutation. I agree, MPL mutation is rare in ET, it's also rare in MF, but if we understand a bit more about targeting with the calreticulin antibody, MPL is the actual receptor that calreticulin binds to. So if we could maybe get that antibody to stop the binding, we may be able to target the MPL mutation, but we haven't got so much information on that. It only affects roughly around 5% of individuals. So Adam, I'm going to turn over to ask you now that question about the variant allele frequency, etc.
Yes, so why are we not doing that routinely? Well, I think the important question with any test that we do in the clinic is, how will that make a difference to the way you manage a patient and the information you give a patient? So in some situations, I'm not sure measuring the JAK2 variant allele frequency does make a great difference, actually. In a patient who's on hydroxycarbamide, for example, it may not be important to monitor the levels. Having said that, I am very much a strong advocate for the application of these types of molecular analyses. And a number of years ago, I pushed very hard to make sure we routinely did report JAK2 variant allele frequency routinely in the laboratory in Oxford, which we've been doing for a number of years now. And I think that is happening more and more. So I think that is coming, and largely that will be available in most labs, I think, over the coming years. The other thing to say is what is not established is, how important it is to do regular monitoring once a year, once every 6. There are certain questions we don't know the answer to. How often should you monitor molecular levels? We don't know. Every 6 months, every year, every 2 years, we don't know that. This is why it's so important to run studies like the MAGIC study, because you can really learn a lot about this. What my take home from that is probably these molecular responses often don't happen very quickly, and sometimes it takes a bit of time. So probably once a year is more than enough, and not everyone needs that doing.
Good answer. I think
And
we
I don't think it's cost is the other thing. I don't think it's cost, because most of the ways people measure JAK2, actually the variant allele frequency is already available. It's just not reported. So there's no additional cost really.
Okay. There's a question about ASXL1 gene. Could we talk about the significance of the ASXL1 gene? So I'll let you think about that one, Adam. And then there's quite a few questions about hydroxycarbamide or hydroxyurea, which I'll just briefly answer. If you stop that medication, there's a question from somebody in Canada, I think, about giving your body a rest and stopping it. You will find that your blood counts will go back up. Patients can sometimes pause interferon and get a prolonged response, and our colleague Jean-Jacques Kelajan has shown that. But for hydroxycarbamide, that's probably not the case. It can increase the risk of skin cancer in both men and women, and it can impair fertility in both men and women. So there have been quite a few questions about that. Can hydroxycarbamide reduce the allele burden? It's been shown in some studies to do that initially, but not in a sustained way. I'm going to go over to you now, Adam, about ASXL1. And there's another question that's just come in about, should MF patients be screened for additional mutations at regular intervals? And if so, how often? Would you mind answering that one as well, please?
Yeah, well, I'll take that one first and then go on to ASXL1. So again, I would go back to what I said before. I'm a strong advocate for the application of these kind of cutting edge molecular tests. But I think we nevertheless still need to use them wisely. And what I mean by that is, we have to, as clinicians and patients, understand why are we doing them? How is that going to make a difference to how we're managing a particular patient? In the case of the more extensive molecular tests, the situation where it particularly makes a difference is at diagnosis. And at diagnosis, once you've made a diagnosis of a particular myeloproliferative neoplasm, the next step is really to accurately risk stratify the disease. So work out what are the complications and the risks that are associated with the disease for any individual patient. And in all the different types of MPN, that involves a mixture of assessing clinical factors like age, blood count parameters and so on, but combined with these molecular abnormalities. And those things can, together, if you combine the clinical and molecular, it gives you a more precise prediction of what the future holds for patients than just doing the clinical factors alone. So I think that's the strongest justification for doing this. But nevertheless, in some situations, it's not a helpful thing. Some patients, for example, do not want to have a detailed discussion about prognosis. And, that's up to them. And of course, if they don't, then I wouldn't routinely check those additional molecular markers. In terms of regularly monitoring things, That we don't yet do in Oxford, apart from in selected cases. So my practice at the moment is to repeat a myeloid gene panel when there's some clinical indication of something changing for the patient. Maybe their blood counts are changing or some new symptoms or something like that. And that I would typically use as a prompt for carrying out an additional molecular analysis. And then looping back to ASXL1 mutation, that is an important mutation to find in persons with particularly myelofibrosis, but also some of the earlier myeloproliferative neoplasms. Some studies have shown that the presence of an ASXL1 mutation does correlate with an increased risk of the disease progressing. However, I don't want anyone to think if they've got an ASXL1 mutation, that inevitably means it's really, really bad news. No, it's much more nuanced than that. You have to take that mutation in the context of presence of other mutations and also in the context of the other clinical factors. So none of these things in isolation are important, but it just adds to the overall information and the precision by which you can give information to the patient.
Okie dokie. Adam, I'm going to come to you with one last question, which has just appeared, which is very pertinent to what you were just saying, but I'm going to first talk about COVID. I don't want to end on COVID because we've done so many of these on COVID. So that if you can look in the Q&A, there's a question from Robert Butchart about prediction of mutations about evolution of disease, which runs very nicely from what you were just talking about. And there is a question that came in before the webinar about, which says, how are MPN patients doing now if they get COVID and being fully jabbed? And then it says, at one time, I recall someone from Guy's saying half the patients in the ICU because of COVID were MPN sufferers. Not true. Definitely not true. And we have very few COVID patients on ITU at the present time, even though there are quite a lot of COVID in the community. Just a reminder that MPN patients are eligible for antiviral treatments. All myelofibrosis patients are eligible, and any ET or PV patients who are taking a drug such as interferon or hydroxycarbamide for their treatment are eligible for antiviral treatment. So if you have a positive COVID swab, you need to speak to your clinical team and you will be referred to a COVID medicines delivery unit. There's a question about shielding and only socializing outside. I think we should be aiming for life to move towards as much towards normal as you are comfortable with, but continue to take precautions. There are a lot of other viruses circulating in the community at this time of year. We've all probably had at least one of them. I certainly have, from my children returning from university. So hopefully we won't need to focus too much on COVID going forward. But at least it did give us the opportunity to connect via these kind of webinars, etc. So Adam, if you wouldn't mind just talking in the last answer we're going to give today about mutations and predicting evolution. We've talked about molecular response predicting, maybe reducing the risk of transformation and altering the course of disease, but now about whether these mutations do predict evolution, if you wouldn't mind.
I think that's exactly what they do. They do predict. You know, I mean, that's essentially what the additional mutations represent. These are all. If you can think back to that little cartoon I showed at the beginning of my talk, in all cases, an additional mutation arises in one individual cell, and then that cell gradually grows. So that's what we mean by evolution, that you know, for the process of going from that one cell that acquires the mutation to that being detectable in a blood test is a process of evolution. And you know, some mutations predict for a more rapid and important kind of process of genetic and clinical evolution in the disease than others. And that's quite well documented now, the exact list of mutations that are strong predictors of evolution of the disease versus those that are not. there are some additional mutations that are essentially pretty, in inverted commas, benign, don't really have an additional impact. And there are others that are much more worrying and can most definitely influence the decisions you make. So hope that answers your question.
I think so. And just to advertise that one of our, thank you so much, Adam. I'm going to hand over to Nona, but one of our next webinars is actually going to be on genomics. So for those who asked all those fascinating questions, or if you have some other unanswered questions, we will be having a session on genomics. So over to you now, Nona.
Well, all I can say again is a huge thank you to both of you. This has been incredibly enlightening and also very exciting for the future of MPNs and treatments. And as patients, we couldn't be more grateful for that. And it's so lovely that we've got so many people from around the world. Just one thing, you know, we do a lot of research or try and fund as much as research as we can from MPN Voice, but we can't do it without donations. And if you've enjoyed this webinar and feel that you can contribute anything, you know, it doesn't matter how little or how much, every penny counts, or dollar or whichever currency it is. And you could do it through the QR code. There's a funny thing come up on the screen. I don't know, we've lost the QR code.
It's coming. If we go to the next slide, yeah.
The previous slide.
There we go.
There it is.
Thank you. That is the QR code. You could also donate through justgiving.com/MPN Voice. If you've got any queries about fundraising or what we're doing or where the money is going to, don't hesitate to contact Maz at info@mpnvoice.org.uk. We look forward to seeing you all again. Next slide, please. And just again, on behalf of MPN Voice, I'd like to say thank you to everyone in our community for your ongoing support. The people that contribute to Health Unlocked, to Facebook, to Twitter, but most importantly, to all the clinicians who generously give up their free time to support this forum. You know, it really is appreciated by every single person, every one of us in the community. So my heartfelt thanks to all of you. We couldn't do without you, and we really look forward to seeing you all again. And I just have to say, stay well and keep safe. Thank you.